Welcome back, everybody. We're going to be looking at Chapter 8, Patterns of Inheritance in our Concepts of Biology OpenStax textbook. We'll have 3 sections in this chapter. Section 8.1, Mendel's Experiments, 8.2, the Laws of Inheritance, and 8.3, Extensions of those Laws of Inheritance. The little brief history, people didn't always understand how traits were inherited from parents to their offspring. All they could really see was that their children or their offspring looked a lot like the parents and seemed to kind of be a blend of both of them, which then 2 theories emerged. The blending theory, where offspring were a straight mix or blend of the parents, and a particulate theory, that traits are inherited as particles, that offspring receive a piece from each parent, and some pieces may hide others. But one of the best known of the time in the 1800s was the blending theory, such as if you mixed a white flower with a purple flower, then you would get kind of an in-between as your offspring, kind of maybe a lavender. We now know that genes are carried on chromosomes and are the basic functional units of heredity, with the ability to be replicated, expressed, and even mutated. Our next slides will cover both sections of the chapter 8.1, Mendel's Experiments, and 8.2, his Laws of Inheritance. So who was Gregor Mendel? He's actually known as the father of genetics. He lived in the 1800s and ended up being very interested in science and nature, but one of the best ways at the time to get a good education was to join the clergy, because there was a lot of money there and he would be able to complete studies there. And so he ended up joining the monastery and was able to conduct his research while there. He used a simple biological system and conducted methodical, quantitative analyses using large sample sizes, which that's a great way to do science, having quantitative data and using large sample sizes in the thousands, and taking good notes as well. His studies revealed how traits were inherited, keeping in mind that in the 1800s we had not yet discovered DNA or chromosomes even. So doing all of this without a basic knowledge of the DNA that we have today or chromosomes is pretty interesting that he was able to accomplish all of that. The postulates that he put forth form what's called classical or Mendelian genetics. We kind of give it that nickname these days. But not all genes are transmitted from parents to offspring according to Mendelian genetics. We've found that there are exceptions to his rules, but Mendel's experiments serve as a really good starting point for thinking about inheritance, and we give him a lot of credit for doing these studies at a time where not a lot was known, and doing science well. So genetics is the study of heredity, and heredity is the passing of traits from parents to offspring. Mendel did start work on both honeybees and pea plants, but luckily he did choose pea plants as his model organism for conducting his genetic studies. They have a lot of different traits, such as being tall or short, that are a very visual trait, having purple and white flowers, round and wrinkled seeds, yellow and green seeds, all very nice visual traits that he could track through generations. Plus, pea plants can be self-fertilized or cross-fertilized. Self-fertilized meaning you can take the pollen from the same plant and fertilize the same plant, or you can take the pollen and fertilize different plants. That's what cross-fertilization means. And there's a picture of a pea plant in this slide with purple blossoms, purple flowers. And Mendel's process for performing crosses included examining flower color. And he selected pea plants as his primary model system. Mendel's work, unfortunately, after he did all these studies, went unnoticed by the scientific community for quite a while, which incorrectly believed at the time that the process of inheritance involved a blending of parental traits that produce an intermediate physical appearance in the offspring. While in some cases that can be true, generally it's not quite true the way that works. This hypothetical process appeared to be correct at the time because of what we now know as continuous variation. Continuous variation is a range of small differences that we see among individuals in a characteristic like human height. It does appear that offspring are a blend of their parents' traits when we look at characteristics that exhibit continuous variation. And Mendel worked instead with traits that show discontinuous variation. Discontinuous variation is variation seen among individuals when each individual shows one of two or very few easily distinguishable traits, such as violet and white flowers, such as a violet flowered plant and a white flowered plant get together, and then all the offspring are either white or either purple, or some of them are purple, some of them are white. Not the blending that people were thinking of at the time, like the white and purple mixing to make kind of a light lavender color. While in some cases, some types of inheritance do work that way, not in discontinuous variation. So here's some other pea plant characteristics. Round and wrinkled seeds, yellow and green, purple and white flowers, inflated and constricted pod shape, yellow and green pod color. Where the flowers are positioned, axial meaning a little lower, terminal at the top of the stems, and stem height, whether they're tall or dwarf, which means short. All of these very nice visual traits that can be studied with pea plants. So we're going to take you through a pea plant experiment as if we were in Mendel's day, and we're going to use blossom color, whether they're purple or white blossoms. So to get ready for the study, Mendel spent a lot of time preparing, and he basically had an area where you would have pea plants growing, and they would only ever be white with their flower color, and another group that were only ever purple. That way he knew they were true breeding parents, the P generation. P means parental. So they're true breeding parents. They only have the ability to make either purple or the other group just white. And once that was done, it took about 2 years, which is very patient work. He cross-pollinated them. So here we have the pollen being transferred to the other flowers. And then he looked at what's called the F1, or first filial generation. Filial means offspring. Now, if the blending hypothesis was right, then we would have expected kind of a light purple flower, but that's not what Mendel got. So in the first filial generation, F1 generation, all flowers, 100% of them, were purple. He didn't just stop there, which is very interesting. He kept going, and this is kind of the question is, okay, where did the white flowers go? Are they gone forever? But he let these purple flowers self-pollinate to make what's called the second filial generation, or the F2 generation. And what do you think he got? Let's take a look. Oops, there we go. So there's our purple and white flower. Our parents, who are true breeding, they mix together and make the F1 generation, who are 100% purple. He allows them in this F1 generation to self-pollinate. And then in our F2 generation, look what we've got. We get 75% or three-fourths of them purple and 25% white. There's our white flowers back again. This is a 3-to-1 ratio here with his numbers. So our blending hypothesis, we can throw that out, and this is discontinuous inheritance here. So what did all of this mean? So Mendel's postulates, or his ideas about inheritance that he published, are as follows: that traits come in alternative versions, such as purple versus white flower color. And that when these genes or traits come in alternative versions or alternative genes, we call those alleles today. So an allele is a version of a gene, such as the gene for flower color comes in 2 alleles, either purple or white. This is due today, we know, because of the different sequence of DNA. The different sequence of the nucleotides A, T, C's, and G's that are located in DNA at a specific locus, or the location of a gene on a chromosome. So remember, A, T, C's, and G's, adenines, thymines, cytosines, and guanines, make up DNA, and it's that specific order of those that makes up a gene. And if you have you could just have one letter off, one nucleotide off, and it could cause the different flower color. It might be more than that for the difference, but could be just a very slight difference, and you'll get a different allele or different version of the gene. So visualizing that, visualize 2 homologous chromosomes, maybe chromosome number 1, and here's one allele for purple flowers and one allele for white flowers. They're at the same locus on the chromosome or location. And the pea plant got one copy from one parent and the other copy from the other parent, and could carry both copies, or could carry the same 2 purple copy alleles or 2 white copy alleles. So once again, traits come in alternative versions called alleles. For each characteristic, an organism will inherit 2 alleles, one from each parent. This is for a diploid organism. As we've already studied, diploid, the prefix means 2. So we have 2 of each chromosome because we get 1 from each parent. And they are homologous and have the same genes on them, but as we see back here, they may have the same genes, but the alleles or versions of those genes might be just slightly different from one another, causing the trait to be just different, either drastically different or slightly different. It's like having 2 editions of an encyclopedia where the definitions of the words inside are not always exactly the same. Another thing, besides figuring out that there are alleles, different alternative versions of genes, and that they get one from each parent, Mendel also discovered that some traits have the ability to mask others. Mask or hide. So for instance, we got our purple flowers and our white flowers. When you combine them together, the old hypothesis, we would expect them to blend into kind of a light purple or lavender color, but that's not what happened, hence I've crossed this off here. Instead, the purple was able to mask or hide the white version. Even if an individual was carrying both alleles, the purple hid the white ones. We call the one who's able to do that the dominant allele. It's the one who is making the functional protein and masking the other alleles. The other one is called the recessive one, the recessive allele. It gets hidden or masked or covered up. And in some cases it's making a different protein or a malfunctioning protein, something that's different from the dominant. In some cases, the dominant is the one that we find in nature most often, and we call that the wild type. Not always, but often. And the recessive one, the malfunctioning one, we would call the mutant, but not always. Then Mundo found there's a difference between how an organism looks and its genetics. So we can describe the physical characteristic of the organism as its phenotype. And I remember that they both start with a pH, physical characteristic, and phenotype, pH, beginning to the word. And then there's the genotype, that's its actual genetic makeup. So for instance, let's look at the purple and white flower example here, that this F1 generation, its phenotype is purple, but its genotype is that it's carrying both a purple and a white allele. We can't see that on the outside here, we just see a purple flower, but on the inside in the cells, we know that it's carrying one of each. So its phenotype can be different from the genotype. For genotypes, we can represent the alleles as letters. So for flower color, because the dominant color is the purple flower, we're going to use a capital P. And then for the white flower, since it's recessive, we're going to use a lowercase p. So the dominant allele gets the capital letter and the recessive allele gets the lowercase letter. So then put that together. For our parent generation, we knew that they were true breeding for their purple flower color. So their genotype would have been big P, big P. For the parental white flowers, we knew they were true breeding, and so they would get lowercase p, lowercase p. So put that together, and we can do little problems like this. Here's the parental purple flower with the parental white flower, pea plants. They mixed together, or bred together, and made the F1 offspring, and so all of them, they were 100% purple with their phenotype, but their genotype was one of each allele, big P, little p. But we know that little p, the allele for the white flower, was being masked or covered up by the dominant allele. Here's another example of that experiment in this figure, using yellow and green peas, and then using Y's for their genotype letters. Albinism is an example of a trait that can be found in many animals, including humans. It means that you're not making pigment, so you have a lighter coloration. It's a recessive condition, recessive allele. So we could choose a letter like little a for albinism, and big A would be normal pigmentation. So this child here looks to be of African descent. The genotype would be little a for this child, meaning that it would have had to have inherited a little a from each parent, though each parent has normal pigmentation. So here's that same diagram, but now we've put in the genotypes. So we've got the phenotypes already listed from before, the purple and white flowers, purple and white to begin, 100% purple in the F1 generation, and then in our F2 generation we get purple and white back again. But now we've got the genotypes listed. All of our F1 generation again are big P, little p, they're all the same. So what is the F2 generation? We know we have a 3:1 phenotypic ratio, but the genotypic ratio could be different as well. So this is where we use Punnett squares to help tell us what these numbers are here, what these percentages or fractions should be. So a Punnett square then can help us track a trait. And so we've got on one side would be the male sperm genotype for that trait that we're tracking. And then the other one, the female or eggs, what they provide, what genotype they provide. And then we just bring them down. So bring the big P down, big P over. So some offspring could possibly be this. Big P over, little p down. Always write the dominant one first. And so some offspring could be this, big P, little p. Again, the eggs are providing little p. Sperm is providing the big P. So again, another big P, little p. And then little p over from the eggs, little p down from the sperm. So little p genotype, that's going to give us white flower. So now we can look at both genotype and phenotype ratios, or you can use fractions as well, and percentages. So I typically like to use percentages and fractions, but sometimes we use ratios also. So let's do the genotype first. So what percent are big P, big P? So that would be 1 out of the 4 boxes, 1 out of 4, 1/4, or 25%. What percent or fraction are big P, little p? Well, that would be 2 out of the 4 boxes, or 1/2, or 50%. And then what percent are little p? That would be 1 here out of the 4 boxes, 1/4, or 25%. So those are our genotypic ratios using just the letters. Pretend you don't have the pictures to look at, but just the letters. The phenotype, we'll need to know what it looks like, so it's good to have the pictures there. So with this, what percent are purple? Well, 3 out of the 4 boxes, 3/4, or 75%. What percent are white? That would be 1 out of the 4 boxes, 1/4, or 25% for white. So notice here that our phenotype and genotype ratios are different. But ratios, fractions, and percentages. So the genotypic ratio then was a 1 to 2 to 1 ratio, while the phenotypic is 3 purple to 1 white, 3 to 1 ratio. So there's different ways to look at that. When an organism for a trait, when they have the same alleles, such as 2 purple alleles, big P, or little p, we call them homozygous for that trait. Homo, that prefix means same. If they get one of each allele, such as big P, little p, we call them heterozygous for that trait. Keeping in mind we have a lot of different traits that make up us as an organism, so for each trait at a time you can be homozygous or heterozygous. So 2 organisms can have the same phenotype but have different genotypes. So how does that work? Well, let's say you have the purple flower once again. You could be big P and be purple, but you can also be big P and be purple. So if you're big P, we call you homozygous dominant. You're carrying that dominant allele. If you're big P, then you're just heterozygous. Hetero means different, so you're carrying one of each. Can't tell by looking at you, as the penguin is saying there. So how would we be able to determine the genotype of an individual with the dominant phenotype? So let's say we have a purple flowered individual. How would we be able to tell that they are actually big P or big P? What would we do? We can do what's called a test cross. This is where we breed that individual who has the dominant phenotype. So take a purple flowered individual, and we would breed them with someone who is homozygous recessive, like the white flower. Anybody who is the white flower, we know for sure they are homozygous recessive or little p for their genotype, because that's the only way you can be white, is to carry both recessive alleles. So we know that all white flowers for these pea plants are little p. So we're taking this individual who we're not quite sure, are they big P or big P, and we're going to mate them with a homozygous recessive individual who we know for sure is little p. Then we're going to see what we get. If we breed them together and all the flowers turn out purple, then it's probably a big chance that this parent was big P. But if we breed them together and we get half and half purple, half white, then we know that that purple flower has to be a carrier of the little p allele. That's the only way that we would be able to get other white flowers here in the offspring. So that's called a test cross. That can be done in many different situations in different breeding programs. So a test cross can be performed to determine whether an organism is expressing a dominant trait is a homozygous for that trait or heterozygous. So here's a test cross using the green and yellow peas example. So from all of this, from all this work that Mendel did, he actually came up with some laws of heredity, not just the postulates there, but some actual laws. And one of those laws is called the law of segregation. Remember, he's doing all of this without really knowing that chromosomes exist or what DNA is. So pretty amazing. And so the law of segregation occurs during meiosis, that the alleles segregate. The word segregate means separate into different gametes. So for instance, if your genotype was big P, you can't give both your big P's to your offspring. You can only give them one because they're going to get the other one from your partner. So both these big P's during meiosis will be pulled apart as the chromosomes are pulled apart. Well, then so are the alleles who are riding on them. They'll be pulled apart and put into separate gametes. Now, if you're big P, for this trait, it's no surprise what's going to be in your gametes. All of your gametes will have big P's. Same thing with the little p's. They separate into little p's. But if you're heterozygous, the big P separates into gametes and the little p separates. And so some of your gametes will be the big P, some will be the little p. So we can visualize that here with a pair of homologous chromosomes, and those chromosomes get pulled apart. Therefore, the alleles must separate into separate gametes during meiosis. Again, Mendel didn't even know genes or DNA existed at the time. So that law of segregation during metaphase 1, remember they all line up, those homologous pairs line up together in the middle of the cell. So we've got mom and dad's chromosome lined up there together, and then remember they will be pulled apart or separated or segregated from each other. But remember, there's those other awesome things going on like crossing over. So all of dad's genes might not be on this sister chromatid here. It might be a mix of both mom and dad. So there's other things at work as well. And it's random segregation during the first division in meiosis. It can lead to a variety of possible different genetic arrangements. Again, the pictures that come from the book that go into the slides are very small, but if you go to the book itself, you can see the pictures bigger, maybe easier to read. So all of this is accomplished during a monohybrid cross. Mono means one. This is when we're tracking one trait at a time, a single characteristic, such as flower color or seed color. But we can do things a little bit deeper here. We can track two traits at a time, which Mendel actually did. Those are called dihybrid crosses, where you're looking at two things at a time, such as seed color and seed shape. So when we look in the picture here, we can see there's both yellow and green seeds, and we can see that some of the seeds are nice and round, but others are wrinkled. So this helped Mendel work out some genetic rules. So a dihybrid cross might look like this. We're using the yellow and green seeds. Yellow is dominant, so that gets a big Y. Green is recessive, gets a little y. Then the round shape, big R for round because it's dominant, little r for wrinkled because it's recessive. So we've got two parents here. This parent is yellow round, and this parent is green and wrinkled. So we just take the genotypes and put them together. Then we make sure they're first true breeding, of course. They're homozygous for their traits. True breeding means homozygous. And then we cross-pollinate them, and then we see what is the F1 generation going to look like. Well, it turned out here that 100% are yellow round. Does that mean our green wrinkled has disappeared forever? No, we know those are the recessive traits, so they're just being hidden or masked for a generation. But we let these self-pollinate, and so in our F2 generation, we get this ratio. 9 out of 16 of them are yellow round, 3 out of 16 green round, 3 out of 16 yellow wrinkled, and 1 out of 16 green wrinkled. A 9 to 3 to 3 to 1 ratio. So what's going on here? If genes are on different chromosomes, how do they assort into their gametes? Do they assort together, such as the dominant traits always stay together? Or do they assort independently? Are things more random and mixed up? So if the like all the dominant ones assorted together, we might just get this. Two choices for our offspring. But if it's more random or independent, then we get this. Things are mixed up a little bit better. So which system explains our data better? Well, we already saw this, so I think the cat is out of the bag here of which one it's going to be. It's not this one. It's like we said, if all of our dominant traits stayed together and the recessive ones stayed together, we would get this. And this is not what we got. Oops, I am in, move my picture. I'm in the way here. So we're not just getting yellow round and wrinkled green. We're getting this over here. We're getting more of a mix where the traits mix up. So this is what a dihybrid cross looks like. All right, so for a dihybrid cross, we need 16 squares instead of just 4 for a monohybrid cross. So the first thing that we do in order to fill this in is we find the allele combinations for what these parents can produce in their gametes. So if you remember the FOIL method back from a math class, you do first, outside, inside, last. So it might not always go in that order, but what we're going to do is find the combination. So big Y could go with big R. Each gamete can only have one of the Y's and one of the R's. So each parent can only give one of those. They can't give both because they're going to get the other one from the other partner. So big Y and big R. Then we can do big Y little r, could be in the gametes. We could do little y big R and a little y little r. So then we list those possibilities across the top. Now the other parent has exactly the same genotype as the first parent. So when we find the allele combinations here, they're exactly the same. Big Y, big R, so on and so forth. So we list those. Then we put them into the boxes. Put your Y's together. Big Y, big Y. Big R goes here. And we keep doing that till we have all of them filled in. Our last box is little y, little y, little r. And this slide is nice enough to also make it visual by showing you what the P actually looks like. So this P with big Y, big R is yellow and round. Where this one here, big Y, little r is yellow and wrinkled. Down here, our last one, little y, little r is green and wrinkled. So then out of 16 squares, 9 are yellow and round. Count them up. Out of 16 squares, 3 are green and round. Here they are. 3 out of 16 are yellow and wrinkled. See if you can find them. Here, here, and here. And then 1 out of 16 is green and wrinkled. So there's our 9 to 3 to 3 to 1 ratio for our dihybrid cross. And Mendel performed these crosses, amazingly enough, using 2 traits at once, going from the parental generation to the F1 to the F2, and sometimes even beyond that. Having big sample sizes, taking good notes, and amazing. So here's that problem with the 16 square Punnett square for the dihybrid cross. If you're looking for a good lesson in how to do dihybrid crosses, go to YouTube and punch up Amoeba Sisters dihybrid cross, and they will take you through it step by step. It explains it very well. All right, so from doing dihybrid crosses and looking at the inheritance of those, Mendel was able to come up with another law of heredity. It's called the law of independent assortment. This means that different genes will separate into gametes independent of one another. That the genes for the green and yellow seeds and the genes for round and wrinkled seeds aren't like following each other together. They're assorting into their different gametes independently of each other, which gives us all that variety, that 9 to 3 to 3 to 1 ratio. Today we know that there are some exceptions to this rule, that it's only true for genes that are on separate chromosomes. Sometimes when genes are really close to one another on the same chromosome, we find them inherited together a lot. usually due to the fact that they cross over together during crossing over, because they're on that same chunk of chromosome together. So we call those linked genes, and they become the exception to the rule of the law of independent assortment. But for genes that are on different chromosomes, it's true for them. And what creates this? Again, metaphase 1, when those chromosomes line up in the middle of the cell during metaphase. Basically, remember how that works. It's random of what side they go on. They're not watching any other chromosome and making sure that they go on the same side all the time. But it's random whether they're on the left or the right. And all 23 pairs of chromosomes are randomly doing this with their partner. Again, linked genes are the exception to this rule. Good thing when Mendel chose his pea plants and these visual traits that we're looking at, that they actually followed this law of independent assortment. Otherwise, it probably could have been very confusing in the time of Mendel to figure this out. So you can trace the genetic events here through meiosis to gamete formation. See the principle of segregation, that both mom and dad's genes have to segregate into separate gametes, and then the law of independent assortment, that it's random the way those genes do so. So a little review. Monohybrid crosses show the law of segregation for a single trait, that each allele segregates into separate gametes, because you can't give your kid both of them. You have to separate them into different gametes. And the law of independent assortment shown by dihybrid crosses with 2 or more traits for genes on separate chromosomes, that they mix into gametes independently of one another. And metaphase 1 is important during meiosis for both of these. Showing how that works with meiosis here. So Mendel chose peas. Luckily, like I said, they are a good plant for genetic research. Relatively simple genetically. Most characters are controlled by a single gene, with each gene having 2 alleles. We now know there's exceptions to all of these rules, and some things, the way they're inherited, are a little more complex than this. Some traits are controlled by more than one gene, but for the traits that Mendel chose, he was pretty lucky that they were all basically a single gene with 2 alleles, one of them being completely dominant over the other, dominant and recessive traits. So it worked out pretty good. And again, he published his findings, but they kind of went unnoticed for quite a long time, till about 1900. It is said that Mendel eventually became an abbot at the monastery. So as an abbot, he would have other duties besides what he was doing with his scientific research. So that might be another reason why some of his information kind of was overlooked at the time, because he took on this other role and probably got very busy. In this section 8.3, we're going to look at the extensions of the laws of inheritance. It's kind of like the beyond Mendel stuff that, okay, Mendel kind of gave us the basics that make up what's called classic or Mendelian genetics, but today we know that there's more complex things also going on. So one of those things happens with snapdragon flowers. If you've seen those planted, you can plant those in Minnesota gardens in the summer if you like. They show what's called incomplete dominance. Now, if Mendel would have chosen these flowers to work with, they would have supported that blending hypothesis, because when you put these two together for incomplete dominance, you have red flowers and white flowers. You mix them together and you get pink, which is a blend of the parents. And then when we mix the pink together in the F1 generation, look at our choices we get. We get 25% of them red, 50% pink, and 25% white, a 1 to 2 to 1 phenotypic ratio. So again, this is kind of a blend. So in incomplete dominance, there are some genes that exhibit this. The heterozygote, that's these guys here in the middle in the F1, they show an intermediate or a blended phenotype. So red and white, in this case, they do make pink. So that is an exception to the rule. There's another type of flower called 4 o'clocks that do this as well. I used to plant those when I was a kid. Red and white, again, make pink, but then if you breed all of the F1, you can get the 3 color choices in the F2 generation. So it's kind of interesting the way that works. So how do we do the Punnett squares with them? There's different ways, but I see a lot of books using this way, where they use superscripts. So we're going to do the trait, or the character is called color in this case, so we're using a C. So all these C's mean the color. And then we're going to pick out the allele. So we have the allele for red and the allele for white, and both of them are kind of dominant together. So they both get a capital letter. So big R is red, big W is white. And then we put them together in the squares, just like we did with the other monohybrid crosses. So this flower with the superscripts of 2 R's is red, and superscript RW is pink. This one too. And then a superscript with 2 W's is white. So again, nobody is completely dominant, nobody's completely recessive. So both of them show through for the phenotype, and you get pink. And again, we get that 1 to 2 to 1 phenotypic ratio here when we do a pink with a pink. Codominance is very, very similar to incomplete dominance. With incomplete dominance, the heterozygote is a blend of both of those traits, like red and white making pink. With codominance, the 2 alleles will affect the phenotype equally and separately. So a good example of this is human blood types. The ABO blood type, the AB actually. Actually, there's a few things going on here that are interesting with blood type. A and B are actually both dominant together. So if you have someone with AB blood type, their red blood cells actually carry both the A and the B, what are called glycoproteins. And they're seen both separately. You can pick out that, oh, there's the A glycoprotein and there's the B glycoprotein. O is actually recessive to both of those. So we have kind of a dominant recessive thing going on here with blood type. But then the AB thing, we have codominance going on. And it's not a blend because you can actually see both the A and the B glycoproteins. A glycoprotein is a protein on the cell surface that has a little sugar group on the end of it. So like in the picture here, these blue things are proteins in the cell membrane, and then the little sugar group sticking off of those. With the ABO blood groups, those are important because our immune systems can recognize those if they're in our bodies and they shouldn't be there. So for rejection of blood donor, that can be a problem. So that's why we type out ABO blood groups in people. This is also a thing called multiple alleles. It's not just one thing or the other, like purple flowers and white flowers, we actually also have a third allele going on here. So we have the A allele, the B allele, and the O allele. So for this, they're going to use superscripts again. And the little I will be for the O blood group because it's recessive. And then the A superscript for the A allele and the B superscript for the B allele. Again, A and B are alleles that are codominant with each other. So you can have red blood cells that have both, and that would result in the AB blood type. And then there's the little I, recessive to both. So what might that look like? So this blood cell has the A glycoprotein, and somebody who has this type A blood could have both A alleles, or they could have an A allele and an O allele. But remember, the O is recessive and gets covered up. Somebody who has the B glycoprotein, as shown in this picture, could be big B, or they could be heterozygous and carry the big B allele and the O allele. Again, the B is dominant, covers up the O, which is recessive. So this is type B blood. Somebody who's type AB blood has to be this, has to have both alleles, both superscripts there, A and B. And so on their red blood cells, it's not a blend of both. You can actually see, oh, there's the A glycoprotein and there's the B glycoprotein. So you see both of them. So that's codominant. And then anybody who's type O blood, they're carrying both recessive alleles. So they get little I for their Punnett square, or their genotype. And we don't see either glycoprotein. So this is also what makes people who are type O universal donor, because they don't have either glycoprotein to alert the immune system that something is there that shouldn't be. Here's a big Punnett square showing all of that. So again, this is an example of multiple alleles. It's an example of dominance and recessive, with the A and B being dominant over O. And then we have codominance going on with the A and the B being codominant with each other. Another example of codominance is sickle cell anemia. So this is the shape of a normal red blood cell, but there is a mutation of a gene, especially in people of African descent is where we see this even more so. The mutation, or the variant for sickle cell, those people have red blood cells who do this. It's called sickling, and it takes on a sickle or a banana shape, which is not very good for carrying oxygen, which is what the job is for red blood cells. And plus it can clog up capillaries. So someone who has sickle cell anemia could have tissue or cell death due to the sickling, especially during exercise. When you are building up a lot of carbon dioxide in your blood, it makes the blood a little more acidic, and the cells tend to do this even more. So they have to be very careful. And so tissue death, cell death can happen because of this. So this is a codominant condition. The actual regular gene, regular allele for normal blood cells is codominant with the sickle cell one. So someone who is heterozygous can actually be making both types of red blood cells. In this case, it's usually not enough to make them sick, because there's enough good red blood cells, so they often don't even know that they're doing this. So imagine someone who is heterozygous getting together with another who is heterozygous, and then of course 1/4 of the time, if we do the Punnett square, they will have a child who has sickle cell anemia and would make all sickle cells. There's another story that goes along with this. You would think, if this is so bad, why does this continue to exist in the African population? The reason behind that is that anybody who's heterozygous, it actually helps them with the malaria parasite that's in Africa. So we'll probably get to that a little bit later when we talk about evolution. Here's another exception to Mendel's rules. It's called sex-linked traits. The genes that are on the sex chromosomes. So remember, females still have the two X's, and the two X's are homologous and have the same genes on them, but maybe different alleles. But if you're a male and you have an X and a Y condition, then they're not homologous. They have different genes. So any gene that's on a X chromosome in a male, they have only one copy of that. And then anything on their Y, only one copy of that, which kind of changes up the inheritance patterns a little bit. This was first discovered in fruit flies, having red and white eyes. So a little reminder about chromosomes. There's our chromosomes, 23 pair. The first 22 pair are all homologous. And so we have two copies of each gene, maybe different alleles, but two copies of each gene. But there's the sex chromosome. So on a male, there's the big X chromosome, having only one copy of each gene, and then the Y, having only one copy there of each of those. So a scientist named Thomas Hunt Morgan was breeding fruit flies with red and white eyes. So he bred true breeding or homozygous red-eyed females with homozygous white-eyed males. So when we put them together, all offspring, didn't matter if they were male or female, were red eyes. So that tells us red is dominant over white eyes. So then when he mated the F1s together, he got 100% red-eyed females, and then the white-eyed males were 50-50. So here's that mating with the Punnett squares. And from back here, where we had our F1 generation, they were all red-eyed flies here. And then we're going to take our red-eyed parent female and our white-eyed parent male, mate them together. So here's their Punnett square. So just as we would have expected, all 100% of those offspring should be red-eyed, despite whether they're male or female. So we just use brown for the color because it doesn't really matter. But let's take the F1s here and mate them. And so we take two big R, little r's, mate them together. What do we get? Well, we get, 3/4 of them. 3 out of the 4 boxes are red, and 1 out of the 4 boxes, or 1/4, are white-eyed. So what's going on here? In humans and other mammals, there's the two sex chromosomes, the X and the Y. Again, 2 X's becomes a female. They have 2 copies then of each gene, but the X and the Y results in the male, and they just have the 1 copy of each. Here we can see from a Punnett square that male and female, 50-50 chance of being a female, XX, 50-50 being a male, XY. So let's do this problem the right way, because what they did back here was a reciprocal cross, where back here, there we go, where they when they mated a certain color of females with males, they got what was expected. But then when they did the reciprocal cross and made the females white-eyed and males red-eyed, they got something totally different. So it only works out if we do it the right way. So for this, we're going to actually have to use superscripts again, and we're going to have to designate whether we are on the X or the Y chromosome, because they found that the gene for red and white eyes is on the X chromosome. So that means the females will have 2 copies, but the males will only have 1 copy for this. So in this case, we have a red-eyed female, putting the big R's up here. The white-eyed male, little r, using superscripts, showing that the female has 2 copies and the male has 1 copy. So then we put it into our Punnett square. And sure, this worked out here where all 100% of the offspring were red eyes. 50-50 male and female, like we would expect. And then 50-50 red eyes, and then white eyes. And we can also see that all of the females then would be heterozygous for their trait, because they got that little r from their dad and the big R from their mom. And then all of the males just had the 1 copy, and they got this big R from mom. So males get their genes that are on their X chromosome from their mother. So then we do the cross of the F1s together. And so we take a female here who is X big R, X little r, mixed with a male who is red-eyed, the big R. And what do we get? Well, take a look at the results here. 100% of the females are red-eyed, but the males 50-50. So that can help explain how those first results, if I go back to this picture, how these were the result. 50% red-eyed males, 50% white-eyed males. Where we probably wouldn't have expected that. But if you do the problem the right way, using the X and Y chromosomes and the superscripts, it actually works out like it should. So when we do any type of Punnett square where the gene is on the X chromosome or the Y chromosome, those are called sex-linked traits, and we have to use superscripts for our genes. Hemophilia, which is a bleeding disorder, is another example of one of these sex-linked recessive disorders in humans. And so regular blood clotting is a big H. Hemophilia is the little h, it's recessive. So this female here is a carrier. She's got the big H for normal blood clotting, but luckily it's dominant over the little h, so she has normal blood clotting. And this male is normal, but only has 1 copy because this is not on the Y, it's only on the X. So we mate these 2 together, and we see that, well, these individuals here, they'll be normal, this X big H, normal, and even this female here, X big H, X little h, is normal blood clotting, but she's a carrier and can have the ability, just like her mother, to pass that on to future offspring. This male here is normal. Notice the male gets anything that's on the X from their mom, because they get the Y from dad. But this male here has picked up the little h from his mother, and therefore would have hemophilia. This example can be seen in the Royal English family from Queen Victoria's time, that she was actually a carrier, like this individual here, this genotype, and passed it on to some of her sons, and it stayed in the family line for a while, at least, until her granddaughter Alexandra married the Tsar Nicholas of Russia, and they had a son, Alexei, who had hemophilia. And during that time, there was a revolution going on in Russia, and their whole family was murdered. But that's kind of a famous story of history with a little genetics mixed in. If you're interested in that, look that one up. Another exception to the rule of Mendel's rules of inheritance would be what we've already learned about this, the X inactivation. We know that since females have 2 X's, one of them randomly gets deactivated and becomes a bar body. And that's why such as tortoiseshell cats carry both colors. So this shows it visually. We have a zygote who is heterozygous, and one of the X chromosomes has the gene for orange, the other X chromosome has the gene for black. And then in the cells, one of them randomly gets deactivated to a bar body. So in this cell, it's expressing the orange gene, but in this cell, it's expressing the black. So then you get that beautiful patch of black and orange and tortoiseshell and calico cats. And again, there's my cat, Bella, who is a beautiful tortoiseshell kitty. Again, linked genes violate the law of independent assortment. When you have genes that are on the same chromosome or really close to one another, then often they cross over together as a chunk and therefore are inherited together much more than the other patterns of inheritance would show. So those are called linked genes. But if genes are on separate chromosomes, then it's more random and they follow those patterns of inheritance like Mendel set forth back in the 1800s. When genes get mixed up, those are called recombinant, like they recombine. And because of this, scientists have been able to use the recombination percentages or frequencies to map out where genes are on a chromosome. There's our reminder of homologous chromosomes with crossover, and the recombinant chromosomes, the sister chromatids, who now have a mix of both mom and dad's genes on them. Epistasis is another rule breaker to Mendel's laws of inheritance. Sometimes one gene will control the expression of another gene. So in this case, we have a dihybrid cross going on here in two separate genes, but one of the genes is for actually making the pigment or laying the pigment down. That would be gene C. Making pigment is big C. Not making the pigment is little c. Then we have kind of the actual color or how much there is, and so big A is for black, little a is for brown. If you get little c though, it doesn't matter whether you have big A or little a. If you get little c, you're not going to lay that pigment down. You're going to be an albino. So in this case, if we follow the dihybrid cross, we don't get that 9:3 to 3:1 ratio. We actually get a 9:3 to 4 ratio because the C gene is in a way kind of controlling the A gene. That it doesn't matter if you have big A or little a. Depends what you get for your C gene. Little c, you're not going to make brown or black. That's called epistasis. One gene can control the expression of another. Epistasis is also seen in the coloration, if you're a dog fan, in Labrador retrievers, and whether they're black or chocolate or yellow. That you've got this other gene, which is gene E, of whether you're laying the pigment down or not. So it doesn't matter if you get the black or the chocolate gene. If you get two little E's, then you're yellow. Polygenic inheritance is another example of violating Mendel's laws, where it gets a little more complex. Poly means many. This one just simply means that it's not just one set of genes that's controlling the trait, that there's many genes that control the trait. So skin color is a great example of polygenic inheritance. Eye color, hair color, weight and height, and even human behaviors can be polygenic. So it's more of a continuum, because you have to count up, well, how many of this gene did I get for skin color. If I got a lot of light ones, then I'm going to be on this end of the spectrum. If I got a lot of dark ones, I'm going to be on this end. Environment can also have an effect, such as the coat of an Arctic fox, or in these hydrangea flowers, the pH of the soil can influence what color they will be. All right, so we've come to the end. Again, there's many more examples of genetics, but those are just a few. So go ahead and look at the questions that come along with the textbook. I encourage you to go through those to check your understanding. And let me know if you have any questions. I hope you enjoyed learning about genetics.